My name is Ann Kennedy, and I am a professor of radiation biology at the University of Pennsylvania School of Medicine. Today I'm going to talk to you about the hazards associated with exposure to the types of radiation encountered by astronauts during space travel. While we're all exposed to low doses of radiation present in our bodies and in the bodies of those around us, and in the air we breathe, the water we drink, and the foods we eat, the radiation encountered during space travel will expose astronauts to higher doses of radiation and more varied types of radiation than those encountered on Earth. Two of the types of radiation which are encountered by astronauts during space travel, that is protons and highly energetic, heavy, charged particles known as HCE particles, are not encountered by people on Earth and are of particular concern for the health of astronauts. Very little is known about the biological effects of these types of radiation. We do, however, know quite a lot about the biologic effects of other types of radiation. The types of radiation of most concern for the production of biological effects are called ionizing radiations. Ionizing radiation has the ability to penetrate deeply in our bodies and cause damage to critical biomolecules in our tissues that are needed to function properly. The deposition of energy from ionizing radiation results in the ejection of electrons from atoms within cells, which then results in the ionization of atoms in our cells. Such ionizations are only the first event in a chain of chemical reactions which lead to the known biologic effects of radiation, some of which you've probably heard of, like the induction of cancer, birth defects, and the production of alterations in the DNA of our germ cells, which can lead to the heritable genetic changes in our offspring. It is expected that the major biological effects of radiation are brought about by changes in the DNA of our cells, which are the basic units of structure and function in biological systems. So measurements of DNA damage figure prominently in radiation biology research. There are many different kinds of radiations encountered during space travel which could produce adverse biological effects in astronauts. Exposure to ionizing radiation during space travel poses some unique hazards, however, primarily due to the presence of relatively large numbers of heavy ions in space. Although we are not exposed to heavy ions on Earth, we can study their biological effects at the NASA Space Radiation Laboratory, known as NSRL, which is a facility at the Brookhaven National Laboratory in Upton, New York. NSRL provides beams of the energetic heavy ions encountered during space travel, which supports studies on the biological effects of these types of radiations. Heavy ions contain different numbers of protons and neutrons, called nucleons, in the nucleus of atoms, and large numbers of these heavy ions are found in outer space. Iron ions, frequently used as the HCE particles, as well as other heavy ions, produce relatively large amounts of DNA damage, as their track width is considerably greater than those which are produced by X and gamma radiations. A major question in space radiation research concerns how the expected biological effects of space radiations can be minimized. Theoretically, it would seem possible to eliminate the effects of heavy ions through shielding methods. Shielding for heavy ion exposure, however, is extremely difficult as it is complicated by nuclear fragmentation, in which the secondary fragments produced from heavy ion interactions can be equally or, in fact, more hazardous to biologic tissues than the original heavy ion. To account for the biological effectiveness of different kinds of radiation, it is necessary to define some of the terms related to it. Radiation doses are expressed in terms of energy absorbed per unit weight, which in turn is related to the number of ionizations produced within a tissue. The radiation dose is the gray, measured as joules per kilogram. A major difference between the various kinds of radiation is the average distance between the ionizing events. Whereas the distance between the ionizing events ranges from a few hundred to a few thousand angstroms for gamma and X-rays, it is only a few angstroms for densely ionizing radiation, such as alpha particles. The linear energy transfer, that is the LET, is a measure of ionization density, and it is expressed in units of electron volts deposited in tissue per micron of track length. Although there is a continuous spectrum of LET values among the known radiations, for practical purposes, radiations are divided into two primary groups: low LET radiation, which includes X-rays, gamma rays, and beta particles, and high LET radiations, which include neutrons and alpha particles. For high LET radiation, the amount of energy deposited per track length is very high compared to the distribution of energy for low LET radiation, in which the ionizations are spaced further apart. The relative biological effectiveness, that is the RBE, is a term used to compare biologic effects for different kinds of radiation. RBE is defined as the ratio of the dose required for a given biological effect with X-rays to the dose required for the same effect with the radiation in question. In general, RBE increases with increasing LET for a number of different biologic effects. The quality factor, Q, is used for purposes of radiation protection and is another LET-dependent factor by which doses are multiplied to account for the varying effectiveness of different kinds of radiation. The major unit of radiation dose which accounts for the biological effectiveness of different kinds of radiation is the sievert. The radiation from HCE particles is high LET radiation, with characteristics much like the radiation from alpha particles to which we are exposed on Earth. We are all regularly exposed to relatively low doses of alpha radiation from sources in our air, food, and water, and cigarette smokers are exposed to considerably higher doses of alpha radiation. Alpha particles have very little penetrating ability, as they expend their energy rapidly in tissues. For example, a 5 MeV alpha particle having a range of 35 microns in tissue will not penetrate below the cornified layer of the skin, and is therefore not an external hazard. If inhaled or ingested, however, an alpha particle can become hazardous, as it may deposit all of its energy within one or two cells. Thus, the amount of local damage can be very great. Unlike alpha radiation, the radiation from HCE particles has the ability to penetrate deeply into our tissue and cause biologic effects in internal organs. HCE particle radiation is thought to be highly effective at inducing cancer and producing other adverse biological effects compared to X-rays. RBEs greater than 20 have been observed for the production of some types of tumors in animals by HCE particles, suggesting that a dose of HCE particle radiation more than 20 times as much cancer in animals is caused as the same dose of low LET radiation. Researchers of the Radiation Effects Team of the National Space Biomedical Research Institute are examining risks of various biologic effects from exposure to space radiations, and we are attempting to develop countermeasures to reduce the risks associated with exposure to space radiations. So what are the radiation risks for space travel? Radiation exposures during space travel may kill cells, weaken the immune system, cause mutations, and have other effects that can lead to cancer, cataracts, cardiovascular and central nervous system injuries, and several other disorders. Through research projects that cut across several program areas, NSBRI investigators are determining the risks associated with various types of radiation for the production of acute effects and the development of malignancies, and whether it's possible to reduce these risks through pharmaceutical and nutritional interventions. Many of the current NSBRI radiation effects team projects are aimed at assessing risks of space radiation-induced adverse effects, such as those leading to heart disease, that is the cardiovascular risk, cancer, and hematological parameters. It is recognized that ionizing radiation has major effects on the cells of the immune and blood-forming systems, with particularly profound effects leading to decreases in the number of circulating granulocytes, also called polymorphonuclear leukocytes, and lymphocytes, as can be observed in the graph showing the changes with time in numbers of circulating blood cells following exposure to a moderate dose of radiation. Several of the current projects involve studies related to the development of pharmaceutical and dietary supplement countermeasures for the cytotoxic effects of space radiation on hematopoietic cells and their progenitor cells, and for the development of space radiation-induced malignancies derived from hematopoietic cells. Another project involves studies of countermeasures for synergism between space radiation and hind limb unloading, which is an animal model system simulating microgravity conditions in loss of white cells, that is lymphocytes. In the future, it is expected that the radiation effects team will be primarily focused on acute effects of high-energy proton radiation of the types and doses which may be encountered during a solar particle event, which is the subject of the short movie produced by Dr. Ron Turner, which you are now seeing. The estimates of doses to be received by astronauts during a solar particle event vary greatly. Currently, it is not expected that the doses during a solar particle event will be sufficiently high to cause radiation-induced mortality, but the radiation-induced prodromal syndrome, which involves nausea, vomiting, and fatigue, may occur, along with radiation-induced adverse skin reactions and hematological changes. The risks of these effects occurring and their association with various potential doses of radiation, which could be received during a solar particle event, have been estimated by Dr. Ron Turner. Data from past solar particle events have been used to result in the estimations of risks for future SPE events. The most important radiation risk for current radiation workers on Earth and for space travelers has been considered to be the risk of developing cancer. So government regulatory guidelines on dose limits for both of these groups of occupationally exposed people have been based on cancer risk estimates. Radiation-induced cancer takes very long time periods to develop. This is called the latency period during which a tumor develops. Most radiation-induced cancers take decades to develop, so they will not be observed within the time frame of most missions. However, radiation-induced malignancies of blood-forming tissues do not take decades to appear. It is known that they can develop within a few years after exposure to radiation. Thus, radiation-induced hematological malignancies, such as leukemia, will be a hazard for space travelers during what are called exploration class missions, which will involve extended periods of time in space. To give you some information about the types of specific research projects conducted by NSBRI researchers, I am going to share some of the data collected on space radiation effects with you. The work of some of the NSBRI-funded investigators has focused on the development of pharmaceutical and nutritional countermeasures which could be effective at reducing the risks of space radiation-induced malignancies. Our own work has focused on the development of antioxidant preparations as potential countermeasures for space radiation-related malignancies and other adverse effects caused by space radiation. We have primarily used 3 different antioxidant preparations in our studies. One of these preparations is L-Cellino Methionine. While Cellino Methionine is not an antioxidant itself, it is considered an important part of the antioxidant defense systems in our bodies, as it plays a major role in maintaining the activities of 2 powerful antioxidant enzymes known as glutathione peroxidase and thioredoxin reductase. A recommended dietary allowance, that is an RDA, has been established for selenium, which is a trace element, but it appears that higher levels of selenium than the RDA are necessary for cancer preventive activities. Cellino Methionine is an organic form of selenium that has been and is currently being used by the National Cancer Institute in large-scale human cancer prevention trials. We have also used Cellino Methionine as part of an antioxidant mixture containing Cellino Methionine, vitamin C and E, alpha-lipoic acid, and N-acetylcysteine. I'm sure you've already heard about the antioxidant properties of vitamins C and E. Alpha-lipoic acid is a powerful antioxidant that is both lipid and water soluble and is often considered to be like a B vitamin. N-acetylcysteine is a small molecular weight thiol which has antioxidant activity itself. It is also a precursor to intracellular glutathione, which is a tripeptide small molecular weight thiol that is a versatile and powerful protector against radiation-induced oxidative damage. We have also used another antioxidant preparation in our studies, which contains the soybean-derived protease inhibitor known as the Bowman-Burke inhibitor. Bowman-Burke inhibitor concentrate, or BBIC, is the form of BBI utilized in our space studies, as BBIC is the form of BBI which has been used in human trials for well over a decade. Both BBI and BBIC have been shown to protect against numerous radiation-induced biologic effects. As described earlier in this presentation, the important biological effects of many types of radiation are brought about by the ionization of atoms within cells, which in turn can lead to the production of free radicals. As approximately 80% of cells is water, oxygen-based free radicals, known as reactive oxygen species, are thought to be particularly important in the production of biological effects from many types of radiation. Therefore, it seems reasonable to assume that antioxidants might be able to interact with the radiation-produced free radicals and eliminate or minimize the biological effects produced by radiation exposures. As antioxidant compounds have very different characteristics, for example, their distribution patterns in the body, solubilities in biologic tissues, abilities to interact with different free radicals, and so forth, it might be expected that the mixtures of different antioxidant compounds could be more effective than expected for a single antioxidant compound acting by itself. Our work has shown that several different agents, all of which are characterized as dietary supplement agents, can affect radiation-induced biologic endpoints, which have included cytotoxicity, the induction of malignant transformation in cells cultured in vitro, and the total antioxidant status of animals, which is a biomarker related to the ability of the animal to handle radiation-induced oxidative stress. In a collaborative study with other NSBRI researchers and members of the radiation effects team, Dr. Alan Gewirtz and Dr. Betsy Sutherland, we observed that an antioxidant mixture called the antioxidant cocktail in the figures shown here could reduce the levels of double-strand DNA damage produced in hematopoietic cells by iron ions. It is generally assumed that radiation-induced double-strand DNA breaks can result in important biologic effects, so a reduction in levels of these DNA lesions would be expected to result in less biologic damage. This is just one example of a collaborative project between radiation effects team members. The NSBRI structure encourages such collaborative projects, and there are numerous other examples of collaborative studies within the radiation effects team, between members of the NSBRI radiation effects team and other NSBRI team members, and between members of the NSBRI radiation effects team and NASA-funded investigators. At this time, NSBRI investigators have shown that the major space radiation-induced biologic effects of concern, that is, cell killing, mutation, and cancer development, can be prevented or minimized. That is to say that proof of principle exists for the potential elimination of space radiation biologic effects, including those produced by HZE particle radiation. It is now clear that many different types of modifying agents exist. These modifying agents have different potential toxicities and side effects. The challenge for NSBRI researchers now is to determine the best possible combinations of modifying agents so that the potential biologic effects from space radiations can be eliminated with the fewest possible toxicities and side effects of the pharmaceutical or nutritional supplement agents utilized. The investigations of the NSBRI Radiation Effects Team are focused on important research questions of particular relevance to both the NSBRI and NASA. NSBRI researchers will soon be focused on space radiation risks and countermeasures related to the acute effects which may occur as a result of astronaut exposure to proton radiation during a solar particle event. As this is of major importance at this time, I will end our discussion with a photo of a particularly prominent solar particle event. Hopefully, the astronauts will not be exposed to the major radiation doses expected from such a solar particle event. As they could be, however, it is hoped that the work of the NSBRI Radiation Effects Team can result in the identification of specific countermeasures that will minimize or prevent the expected biological effects.